Welcome and greetings to this Altasciences webinar. My name is Jean-Nicholas Mess and I am Principal Research Scientist at Altasciences. The goal of this presentation is to describe the mass spectrometry-based approach we have developed for the quantitation of passive immunotherapeutic agents, specifically monoclonal neutralizing antibodies against SARS-CoV-2, the virus responsible for COVID-19. The approach is selective, precise, and accurate, yet it is generic and easily adaptable to a variety of neutralizing antibodies. Passive immunity is achieved when a person is given already made antibodies, rather than producing their own through the immune system. It is known to provide immediate but transient protection. An example of natural passive immunity is breastfeeding, when a newborn receives a mother's antibodies through breast milk. Starting in the early 1900s, serotherapy from convalescent patients was used as treatment and prophylactic agent for infectious diseases such as measles. Serotherapy for COVID-19 is still being studied, and it is not routinely recommended. An attractive alternative to serotherapy is the use of neutralizing monoclonal antibodies. Such antibodies can be cloned from the B-cell repertoire of recovered patients. They can be produced rapidly, safely, and in large volumes. As of July 2020, 8 neutralizing antibodies against SARS-CoV-2 entered clinical evaluation. Most of them target the virus spike protein RBD domain, which prevents its interaction with the host cell ACE2 receptor. The virus can no longer be internalized and replicated, stopping the infection process. As for any other therapies, quantitative bioanalysis plays a central role in the drug approval process, as reliable pharmacokinetic data on drug exposure is required. Using mass spectrometry, bioanalysis of biologics such as antibodies is commonly performed using a bottom-up approach. Samples can be prepared using different procedures, from generic to very selective. Proteins are then digested with trypsin, where a peptide unique to the protein of interest is used as surrogate for quantitation. Mass spectrometry-based assay offers several advantages, such as rapid method development. It's also very selective based on the uniqueness of the surrogate peptide used, and it can be multiplexed to quantify multiple analytes in concert. Our goal at Altasciences was to establish an LC-MS-based workflow for the quantitation of neutralizing monoclonal antibodies to SARS-CoV-2 that are currently under investigation for the treatment or prevention of COVID-19. Most importantly, the assay had to be generic and easily adaptable to different antibodies to ensure quick turnaround times between assay development, validation, and sample analysis. In order to develop an approach that is selective, precise, and accurate, key assay parameters such as choice of sample preparation, selection of surrogate peptide, and digestion condition must be considered early on during assay development. A sample preparation upstream to LC-MS offers many options of different complexity. Illustrated here are a few typical approaches used for the quantitation of monoclonal antibodies. Immunocapture, using highly selective anti-ID, was incompatible with our objective due to the time constraint of raising such critical reagents. Protein pull-down using immobilized spike protein, which is the therapeutic target of the neutralizing antibodies, was also investigated, but considering that the ongoing clinical programs are on COVID-19 positive patients, we had concern that circulating viral particle or newly circulating antibodies produced as part of a building up immune response could interfere with the assay. The two approaches that grasped our attention were a simple protein G pull down and direct digestion, as they do not rely on complex affinity reagents. They are generic and can be applied to any neutralizing antibodies. Both sample preparation approaches were tested in parallel. Although sensitivity was comparable, protein G pull-down was definitely more selective, which simplified chromatographic separation at the same time. Protein G pull-down provided a nice balance between sensitivity and selectivity, while remaining generic and applicable to other antibodies. A fundamental aspect of mass spectrometry-based quantitation is the selection of a unique surrogate peptide. Antibodies from the IgG family share more than 85% sequence identity, mainly due to the constant regions of their light and heavy chains. Differences in antibody sequences are found in their variable regions, more specifically within the short CDR sequences, which are responsible for antigen binding. Peptides derived from the CDRs have the highest likelihood of being unique and selective to the antibody of interest. But that being said, monitoring a peptide derived from a CDR does not guarantee specificity, especially considering that protein G pull-down will co-extract all endogenous IgGs and that some CDR sequences could be shared by multiple antibodies. As illustrated here, for neutralizing antibody, only one peptide derived from the second CDR and adjacent framework region from the heavy chain was unique, specific, and allowed for the detection of the targeted LLOQ. Other CDR peptides monitored were detected endogenously at different intensities and were not specific to the neutralizing antibody of interest. Another key parameter to optimize is protein digestion, which is a balance between speed, efficiency, and reproducibility. Reproducibility is the most critical characteristic as it dictates the assay precision. Adding to the challenge, variability in the digestion process is typically not corrected by an internal standard. Commonly used internal standards are heavy labeled synthetic peptide, which are added post-digestion, and therefore will not correct for differences in digestion yield. Silac proteins will be the ideal solution, but they are rarely available to support accelerated clinical programs and are time-consuming to produce. Two commonly used trypsin digestion protocols were evaluated in parallel for precision and accuracy. One involves a quick 2 hours trypsin digestion at an elevated temperature of 60 degrees, while the other is more conservative and involves an overnight digestion at 37 degrees. Although similar digestion yields between the two protocols were observed in screening experiment, precision and accuracy of the assay was definitely superior when the samples were digested overnight at 37 degrees, as most quality control samples were in a range of plus minus 10% deviation. So here is an overview of the finalized workflow. The samples are first diluted 40-fold in a 1% Triton solution to inactivate the SARS-CoV-2 virus and make the samples safer to handle. Pull down is performed using protein G-coated magnetic particles for 90 minutes. The process is automated on our Thermo Kingfisher Flex for both throughput and consistency. Following protein purification, the samples are reduced, alkylated, and digested overnight with trypsin to ensure reproducibility. The resultant peptides are analyzed using a Shimadzu Nexera UHPLC coupled to a SciEx 5500 triple quad with typical run times below 4 minutes. To this date, the approach has been applied to 3 different neutralizing monoclonal antibodies, 2 of which have been combined into a multiplexed assay later during the progression of the clinical programs. These assays have all been validated as per recent regulatory guidance and used for the analysis of more than 1,000 samples to support phase 1 to phase 3 clinical trials. So leveraging our experience and this optimized workflow, a Bioanalytical assay for a neutralizing monoclonal antibodies against SARS-CoV-2 can be developed and qualified in 3 to 5 weeks. The rate limiting step is really the synthesis of heavy labeled peptides. The sooner we have access to the antibody protein sequence, the sooner the heavy labeled peptides can be ordered and the method development initiated. Following method development and qualification, the assay can be validated typically within 2 weeks and be ready for sample analysis. We are therefore looking at an overall 5 to 7 weeks time frame between study kickoff and method development to sample analysis. In conclusion, this presentation illustrated how mass spectrometry can be used for the quantitation of neutralizing monoclonal antibodies against SARS-CoV-2. The methods are precise and accurate, they're selective, and can be multiplexed. The developed workflow can be applied to a variety of neutralizing SARS-CoV-2 monoclonal antibodies with minimal adjustments. The approach is currently implemented in our laboratory to support sample analysis for ongoing phase 1 to phase 3 clinical programs for 3 different neutralizing monoclonal antibodies. So on this, I would like to say a very big thank you to my colleagues at Altasciences, as this was a tremendous team effort. On the method development side, I would like to thank Jean-François, Philippe, and Kevork for their dedication and scientific contribution. Many thanks as well to Julie and her team, Jose and Anaita for the successful management and execution of the assay validations and sample analysis. And thank you for attending this webinar. Should you have any questions, comments, or if you would like to discuss our capabilities, please feel free to send me an email at the address you see on your screen. And don't forget to visit our website, altasciences.com. Thank you all. Stay safe.